A child is born dead. The mother hemorrhages. Blood loss, catastrophic. Her husband steps forward. Same blood type, tested, confirmed compatible. The transfusion begins, and her body convulses. Fever spikes to 104. Blood pressure plummets. Through the microscope, her plasma is destroying his red blood cells, ripping them apart like paper in a shredder. They were compatible. The test said they were compatible. The year was 1937. Karl Landsteiner, already a Nobel laureate for discovering the ABO blood system, sat in his laboratory at the Rockefeller Institute, staring at a medical ghost. Something was missing from the map. He turned to an unlikely collaborator, Alexander Wiener, a young serologist with a talent for animal models. Together, they began injecting rabbits with blood from rhesus macaque monkeys, small brown-furred primates common in laboratory colonies. The rabbits produced antibodies. When Landsteiner tested those antibodies against human blood samples, something extraordinary happened. Some human samples clumped. Others did not. Roughly 85% of humans carried the same surface protein as the rhesus monkeys. 15% lacked it entirely. They called it the rhesus factor. Rho positive for those who had it. Rough negative for those who did not. And in that division, binary, absolute carved into the genome, lay the answer to the stillborn child. Here is what they uncovered. The ROS system is not a single protein, but a constellation. Approximately 48 antigens, encoded by 2 adjacent genes on the short arm of chromosome 1. A genomic address labeled 1p34.1 to 1p36. The genes are called RHD and RHCE. Siblings, written side by side in the helix. RHD produces the D antigen, the protein that sparked the rabbit antibodies, the marker that links us molecularly to our primate cousins. Rositive individuals carry at least one functional copy of RHD. They manufacture the D antigen and display it like a flag on every red blood cell. More than 30 different epitopes, molecular footholds where antibodies can bind. Why so complex? Why would evolution build a protein with 30 distinct binding sites if it were functionally simple? A mere marker, a biological barcode. The architecture suggests importance. Proteins this elaborate are typically conserved because they do something. RIF-negative individuals carry a deletion. The RHD gene is simply gone, erased. A blank space in the genetic text where a chapter used to be. Not a broken gene. Not a mutation that garbles the code. An absence. This is the first peculiarity. Evolution rarely deletes. Genes break, mutate, decay into pseudogenes. But clean excisions, entire chapters removed from the genome, are uncommon. They suggest purpose. It should have vanished. Generation 1, 15% frequency. Generation 100, 12%. Generation 500, 5%. Generation 1000, extinction. The mathematics are unambiguous. Deleterious alleles decline. They do not persist. And yet, the duplication event that split RHD and RHCE apart occurred approximately 8.5 million years ago, in a common ancestor of humans, chimpanzees, and gorillas. For nearly all of that time, the D antigen has been maintained, conserved, passed down. And then, roughly 35,000 years ago, a blink in evolutionary time, the deletion appeared in a subset of anatomically modern humans. It should have vanished. Consider the logic of natural selection. If you are RS negative, your first child with an RS positive partner may survive. Your immune system has not yet encountered the D antigen. No antibodies exist, but during birth, fetal blood crosses into your circulation. Your immune system recognizes the foreign protein. It mounts a response. Memory cells form. Your second pregnancy becomes a war. Anti-D antibodies flood the placenta. They coat the fetal red blood cells. Macrophages in the fetal spleen recognize the antibody tags and begin destroying the infant's blood supply. Anemia, jaundice, hydrops fetalis, where the fetus swells with fluid, organs failing. Before the invention of exchange transfusions and immunoglobulin in the 1960s, hemolytic disease killed or permanently damaged tens of thousands of infants every year. This is what biologists call a deleterious trait, a genetic variant that reduces reproductive fitness. Under normal circumstances, frequency-dependent selection should drive our negativity toward extinction. The rarer it becomes, the fewer incompatible pregnancies occur. But it should still decline generation by generation until it disappears entirely. And yet, here's the global distribution as measured by mid-20th century blood surveys. Among white Americans and Europeans, 15% Rh negative. Among African Americans and sub-Saharan populations, 8% Rh negative. Among East Asian populations, Han Chinese, Japanese, Korean, 1% Rh negative. The gradient is not random. It follows geography. It follows ancestry. And it peaks, dramatically, inexplicably, in a single isolated region of Europe. And then you reach a place where the numbers break, where the frequency does not decline gently from 15% to 10 to 5%, where it spikes. 47.2%. Say that number aloud. 47.2%. Not in a small tribe. Not in 100 individuals. In entire valleys, entire provinces spanning 3 million people. The Basque Country. 7 provinces straddling the Pyrenees between France and Spain. And in some villages with churches older than nations, with gravestones carved in a language no linguist can trace, nearly half the population lacks the rhesus antigen. The Basques are not merely statistically unusual, they are linguistically isolated. Their language, Euskara, has no demonstrable relationship to any other language family on Earth. Not Indo-European, not Semitic, not Uralic. Linguists have traced every other European tongue back to common ancestral roots, but Euskara stands alone, a language isolate. Some have speculated it is a remnant of pre-Indo-European speech, preserved in the mountains while waves of migration swept across the continent. Others suggest deeper origins, a Paleolithic substrate, echoing back to the first humans who painted bison on cave walls at Altamira. The genetics mirror the linguistics. Basque populations show reduced genetic diversity compared to neighboring regions, consistent with a founder effect. A small population that remained reproductively isolated, amplifying whatever traits they carried. But that does not explain why the Rn-negative deletion survived at such extreme frequency. Isolation might preserve a rare trait. It does not favor it. Something else is operating here. In 1945, a physician named Echeverri, working in Argentina, noted the high frequency of the Rh negative variant in Basque immigrants. He published his findings in a regional medical journal. The paper was largely ignored. It should not have been. Because Echeverri had identified what evolutionary biologists would later call the Basque paradox, an allele that causes infant mortality, persisting at high frequency in an isolated population for 35,000 years with no obvious selective advantage. By all rights, it should not exist. And yet when you test Basque blood, when you run the genotyping assays and count the deletions, the signal is unmistakable. The absence is louder than the presence. And in evolutionary biology, when something that should be silent, something that kills children, that violates frequency-dependent selection, that contradicts every model of genetic drift, when that thing screams across 35,000 years at 47% frequency, you stop theorizing. You listen. Landsteiner did not live to see the paradox fully articulated. He died in 1943, 6 years after his discovery, still believing the Rh factor was a straightforward extension of blood group immunology. But the questions he opened have only deepened. Why does the human immune system attack its own offspring over a protein shared with monkeys? Why does the deletion persist when it should be lethal? And why, of all the places on Earth, does it concentrate in the genetic refuge of a people whose language remembers no origin? We have established the paradox. Now we must descend into the mechanism. We must look not at blood types as discrete categories, but as molecular territories, surfaces covered in proteins, each protein a word in a biochemical language that your immune system reads with absolute fluency. And we must examine the 8% of your genome that is not yours at all. The viral ruins, the ancient code, the genetic fossils of infections that occurred millions of years before humans existed. Because the route story is not only about a deleted gene. It is about what happens when the human body encounters foreign genetic material and chooses, cell by cell, what to keep and what to kill. Let us be precise about what happens during an R incompatibility pregnancy. Precision is not cold. Precision is the only way to understand the violence. A woman SIC negative, genotype DD, conceives with an SIC man. If he carries 2 copies of the RHD gene, genotype DD, the fetus will be RE-testative, guaranteed. If he is heterozygous D, died, there is a 50% chance. Assume the fetus is positive. For most of the pregnancy, the placenta functions as a barrier. Fetal and maternal blood do not directly mix. Nutrients and waste exchange across membranes, but cells remain separated. But birth is not gentle. During labor, particularly during placental separation, fetal blood cells breach the barrier. Red blood cells carrying the D antigen enter the mother's circulation. Our immune system has never encountered this protein. It is foreign, structurally distinct, and her body does what 3.5 billion years of evolution has trained it to do. It responds. B lymphocytes in her spleen recognize the D antigen. They bind to it, internalize fragments, present them to T helper cells. A cascade of signals initiates. The B cells proliferate, differentiate. Some become plasma cells, factories churning out antibodies, specifically immunoglobulin G. EG is small, monomeric, designed to cross the placental barrier. The first pregnancy may end without incident. The immune response takes weeks to fully develop. The child is born before antibodies reach dangerous concentrations. But memory persists. Memory B cells lodge in the bone marrow, the lymph nodes, the spleen. They wait. And when the woman becomes pregnant again with another RO-positive fetus, the response is not gradual. It is immediate. Within days, antibodies flood the bloodstream. They cross the placenta. They encounter fetal red blood cells and lock onto the D antigen with exquisite specificity. Up to 30 different epitopes, different three-dimensional surfaces on the protein where antibodies can bind. Each red blood cell in an RF-positive fetus carries thousands of these antigens. When antibodies coat the surface, they act as molecular flags. They signal this cell is condemned. Macrophages in the fetal spleen, large phagocytic cells whose function is to consume debris and pathogens, recognize the antibody coating. They engulf the red blood cells, digest them. One by one, the fetus loses the capacity to carry oxygen. The medical term is erythroblastosis fetalis. The old term, still used in textbooks, is hemolytic disease of the newborn. The symptoms escalate in stages. First, anemia. The fetal bone marrow struggles to replace what is being destroyed. Second, jaundice. Bilirubin, the yellow pigment of hemoglobin breakdown, accumulates faster than the immature liver can process it. The infant's skin yellows. Third, kernicterus. The toxin crosses the blood-brain barrier. It deposits in the basal ganglia. Permanent brain damage. Cerebral palsy. Deafness. Fourth, hydrops fatalis, heart failure, fluid leaking into the abdomen, the lungs, the space around the heart. The fetus swells. Fifth, silence. Before 1960, before the development of raw immunoglobulin, a prophylactic injection that prevents maternal sensitization, hemolytic disease was a leading cause of perinatal mortality in developed nations. Tens of thousands of infants per year. Tens of thousands of families watching their second child turn yellow, then silent. Tens of thousands of women whose bodies waged war against their own pregnancies. And evolution, blind, calculating, optimizing for the population, not the individual, did not stop it. Most were second or later pregnancies. The pattern was recognized by midwives long before Landsteiner named it. A woman's first child would be healthy. The second might be jaundiced. The third, stillborn. Families learned to fear the progression. And yet, despite this relentless culling, despite evolution's clear penalty for our incompatibility, the deletion persisted. The Ross system is not simple. Landsteiner's original discovery identified the D antigen, but decades of serological research revealed the full architecture. The RHD gene and its sibling, RHCE, encode a family of approximately 48 distinct antigens. They are transmembrane proteins embedded in the red blood cell membrane, their loops and domains exposed to the exterior. The D antigen alone presents more than 30 epitopes, molecular surfaces where antibodies can attach. Different populations carry different variants. Some D antigens are weak, expressing fewer copies per cell. Some are partial, missing specific epitopes. The complexity suggests functional importance. Proteins this elaborate, this conserved across primates, typically serve critical roles. They are not decorative. And yet, the deletion of RHD, the complete absence of the D antigen, produces no obvious physiological defect in Rh-negative individuals. They live normal lives. They reproduce. Their blood carries oxygen with the same efficiency as Rh-positive cells. The absence costs nothing, except, of course, the children. To map the deletion with precision, geneticists in the early 21st century turned to molecular tools. Tachman quantitative PCR, a technique that uses fluorescent probes to detect specific DNA sequences, allowed researchers to genotype the RHD gene in large populations. The assay is binary. If the gene is present, the probe binds and fluoresces. If the gene is deleted, no signal. Fluorescence in situ hybridization, or fiber fish, stretched individual DNA molecules across slides and tagged them with colored markers. Researchers could count the number of RHD copies directly, visualizing duplications or confirming deletions at the single chromosome level. These technologies reveal that the R-negative phenotype in Caucasians is almost universally the result of a complete RHD deletion, not a point mutation, not a frame shift. The entire gene, approximately 70 kilobases of DNA, is simply absent. In African populations, the genetics are more complex. Some individuals carry an inactive RHD pseudogene. Others have hybrid genes, fusions between RHD and RHCE. The phenotype is the same, no D antigen, but the mechanism differs. This suggests independent evolutionary events. Multiple origins for the Rh-negative trait, but the highest frequency, the most extreme concentration, remains locked in one location, the Franco-Cantabrian region, the Basque Country, a narrow strip of land where the Pyrenees Mountains descend into the Atlantic. 7 provinces, valleys separated by steep ridges, limestone caves where Paleolithic humans painted aurochs 40,000 years ago. Population surveys conducted in the mid-20th century revealed something that should not exist: In some villages, the RHD deletion reached 47.2%. Read that number again. 47.2%. Not 15%, like the rest of Europe. Not 20%, which would already be anomalous. 47.2%. Nearly half the population lacks a gene that 85% of humanity carries. This is not a founder effect gone slightly wrong. This is an evolutionary impossibility that has been stable for 35,000 years. Pause and consider the improbability. An allele that kills children, maintained at near 50% frequency in a population small enough to experience genetic drift over 35,000 years. Frequency-dependent selection should not permit this. Whenever an Rh-negative woman mates with an Rh-positive man, which should happen more often as Rh-negative becomes rarer, her reproductive fitness drops. She loses second and third pregnancies. Her lineage contracts. The mathematics predict extinction, and yet the data say persistence, not just survival, thriving. The Basque language, Euskara, reinforces the mystery. It is agglutinative, ergative, absolutive, with no clear relatives. Linguists have proposed connections to Caucasian languages, to Iberian, to pre-Indo-European substrates, but none have withstood scrutiny. A language with no family, a people with no genetic parallel, a deletion that refuses to die. Theories proliferated. Some argued that the Basques were descendants of the first Homo sapiens to enter Europe, Cro-Magnon hunter-gatherers who arrived 45,000 years ago and remained isolated as later waves of Neolithic farmers spread across the continent. Others pointed to unique cultural practices, endogamy, strict marriage within the community that might have preserved rare alleles. Still others whispered of something older, something unexplained. Because here is the disturbing corollary. If the RHD deletion is genuinely deleterious, if it provides no benefit and only costs, then its high frequency in the Basques is not just improbable, it is impossible. Unless the deletion is not the mutation. Unless what we call normal, the presence of the rhesus antigen, the protein that links us to monkeys, is the anomaly. This is the thought that haunted researchers in the 1970s and 80s as genetic data accumulated and the Basque paradox sharpened into focus. What if Rh-negative blood is not a loss but a preservation? What if the immune war between Rh-negative mothers and Rh-positive fetuses is not a malfunction, but a border defense, a biological recognition that something fundamental has diverged? The hypothesis sounds absurd. It flirts with fringe theories, with claims of alien genetics and lost civilizations. But strip away the pseudoscience, and a legitimate question remains. Why does evolution tolerate an allele that violates the first law of biology? Preserve the offspring in the one population whose genetic and linguistic isolation suggests they may carry a signal from humanity's deep past. We have examined the mechanism. We have measured the frequencies. Now we must ask, what else does our status affect? Because the more researchers looked beyond blood compatibility, the more anomalies appeared. Psychological differences, infections, susceptibilities, behavioral traits. The RHD gene does something. Its absence echoes through the body in ways that transfusion medicine never predicted, and to understand why, we must turn away from blood cells entirely. We must look at the other 8% of your genome, the part that used to be a virus. There is a moment in every biology classroom when the teacher presents the double helix and declares, "This is your genetic code. This is what makes you human." It is a beautiful lie, or rather, it is an incomplete truth. Because if you could sequence every chromosome in your body, if you could read all 3.2 billion base pairs of your DNA, you would discover something unsettling. Approximately 8% of your genome is not human. It is viral, not an active infection, not a pathogen replicating in your cells at this moment. These are fossils, genetic ruins, the degraded remnants of retroviruses that infected your ancestors millions of years ago. Not you personally, not your parents, but the early primates from which you descend, and through a molecular accident inscribed themselves into the germline, into sperm, into eggs, into every cell of every descendant forever. They are called human endogenous retroviruses, HERVs, and you carry approximately 203,000 copies of them. Think of your genome as a palimpsest. In medieval monasteries, when parchment was scarce, scribes would scrape the ink from old manuscripts and write new text over the faded words. Layers of writing, one atop another. The original text remained, ghostly, partial, illegible in places, but it was still there underneath. Your DNA is that parchment. The primary text, the genes that encode hemoglobin, neurons, bone, was written by natural selection over millions of years. But layered beneath and between those instructions, in the spaces we once called junk DNA, are older inscriptions. Viral ink. Most of it is faded beyond recognition. Fragments of reverse transcriptase genes. Truncated envelope proteins. Sequences so degraded that only sophisticated algorithms can detect their viral origin, but some remain startlingly intact. A full-length HERV element is approximately 9.5 kilobases long, 9,500 base pairs of DNA. It contains 4 essential genes, the same architecture found in active retroviruses like HIV. GAG encodes structural proteins for the viral core. PRO encodes a protease, an enzyme that cleaves proteins into functional units. POL encodes reverse transcriptase and integrase, the machinery that converts viral RNA into DNA and stitches it into the host chromosome. ENVin encodes envelope proteins, the surface spikes that allow the virus to fuse with cells. These are not abstract relics. These are functional blueprints, preserved for tens of millions of years, because in some cases, in ways that still astonish virologists, your body learned to use them. The mechanism by which a virus becomes permanent is called horizontal gene transfer. Horizontal, because it moves genetic material sideways between organisms across species boundaries, rather than vertically from parent to offspring. It is common in bacteria. A bacterium acquires antibiotic resistance not by waiting for a mutation, but by absorbing a plasmid, a circular piece of DNA, from a neighbor. Instant evolution. Copy-paste genetics. In complex organisms like mammals, horizontal transfer is rarer, more violent. It requires a virus. Retroviruses are parasites with a peculiar life cycle. Unlike DNA viruses, which commandeer the nucleus and force the cell to replicate their genetic code, retroviruses carry their instructions in RNA. When they infect a cell, they deploy reverse transcriptase, an enzyme that transcribes RNA backward into DNA. This is biochemically perverse. Information in biology is supposed to flow one direction, DNA to RNA to protein. The central dogma. Retroviruses reverse it. Once the viral RNA has been converted to DNA, the integrase enzyme splices it directly into the host chromosome, not into a temporary location, not into the cytoplasm where it might degrade, into the genome itself. Permanent integration. Now imagine this happens not in a liver cell or a skin cell, cells that will die with you, but in a germ cell, a sperm precursor, an egg. The viral DNA integrates into chromosome 7 or chromosome 19, or in some cases chromosome 1. The germ cell survives. It matures. It participates in fertilization. And every cell in the resulting embryo carries the viral sequence. It is no longer an infection. It is inheritance. This has happened repeatedly throughout mammalian evolution. The oldest HERVs in the human genome are estimated to have integrated 90 to 100 million years ago, long before the primate lineage diverged. We share these sequences with lemurs, with tree shrews, with the earliest placental mammals. Others are younger. HERV-K, one of the most intact families, likely integrated within the last 30 million years. Some HERV-K insertions are human-specific, meaning they occurred after our split from chimpanzees less than 7 million years ago. And then there are the polymorphic insertions. HERV sequences present in some humans but not others. The virus is still writing. For decades, biologists dismissed HERVs as evolutionary junk, parasitic DNA, selfish genes that replicated within the genome without providing benefit. But that framework collapsed in the late 1990s, when researchers discovered that one HERV gene, specifically the envelope protein of HERV-WV, was not silent. It was active, and it was essential for human reproduction. The protein, renamed syncytin, is expressed in the placenta. It allows trophoblast cells to fuse together, forming the syncytia trophoblast, the outer layer of the placenta that mediates nutrient exchange between mother and fetus. Without syncytin, there is no functional placenta. Without a placenta, there is no live birth. Viviparous mammals exist because, approximately 25 million years ago, an ancestral mammal was infected by a retrovirus, and instead of dying, it domesticated the viral envelope protein and repurposed it for embryonic development. We are here because of a virus. This is the first pillar of the viral pattern. Your genome is not a sealed system. It is an archive of genetic invasions, most neutralized, some weaponized. You are, in a literal biochemical sense, part virus. Now for the second pillar. Your immune system knows this. Immune tolerance is one of the most delicate calibrations in biology. T cells are trained in the thymus to distinguish self from non-self. Cells displaying self-proteins, antigens derived from your own genome, are ignored. Cells displaying foreign antigens are destroyed. But HERVs blur the boundary. When a HERV protein is expressed, whether through normal cellular activity or through aberrant reactivation, it presents as almost self. The amino acid sequence is close to human proteins but not identical. The immune system hesitates. Sometimes it attacks. There is mounting evidence that certain autoimmune diseases, multiple sclerosis, schizophrenia, type 1 diabetes, correlate with the activation of specific HERV families. In multiple sclerosis, the HERV-W envelope protein triggers an inflammatory cascade that damages myelin, the insulation around nerve fibers. In schizophrenia, HERV expression in the brain during adolescence may dysregulate synaptic pruning. These are not infections in the traditional sense. The viruses are endogenous, already inside you, woven into the chromosomes you inherited from your parents. But under the right conditions, stress, inflammation, other infections acting as triggers, they wake. And your immune system, trained to eliminate foreign genetic material, begins destroying your own tissues because it cannot distinguish the viral remnants from the human architecture built around them. Now, return to the Ross story. An Rh-negative mother, an RL-positive fetus, the immune system's violent rejection of a protein that should be recognized as offspring. The question we have been circling: why does the body treat the D antigen, a primate-conserved protein, present in 85% of humans, as foreign? The answer may lie in how the immune system defines foreign in the first place. If your genome is 8% viral, if the line between you and ancient invader is already blurred, then what happens when a mother and child differ not merely in blood type, but in the deeper genetic context surrounding that blood type? What if ROS incompatibility is not an isolated immunological error, but a signal of broader genetic divergence? What if the anti-D antibodies are not malfunctioning, but functioning with extraordinary precision, identifying a molecular signature that says, This cell lineage is not entirely compatible with mine. This is speculation. This is the edge of known science, where data becomes interpretation and interpretation flirts with hypothesis. But it is grounded in a principle that geneticists have come to accept over the last 2 decades. Humans are not a genetically uniform species. We carry different HERV insertions, different polymorphisms, different deletions. The RHD deletion is one of many, but it is the only one that triggers an immune war between mother and child, and it peaks in the population, the Basques, whose genetic isolation suggests they may have avoided or resisted some of the horizontal transfers that swept through other European groups. We stand at a threshold. Behind us, the mechanics of blood incompatibility, the geography of a deletion, the viral fossils embedded in our chromosomes. Ahead, the anomalies that do not fit, the data that whisper of something deeper than blood type. Because our status does not only affect transfusions and pregnancies, it affects psychology, infection, behavior, and no one knows why. Stop. Something is wrong. The Basque frequency, 47.2%, maintained for 35,000 years, despite hemolytic disease, despite infant mortality, despite every mathematical model predicting extinction. It has not declined. It has not vanished. It has persisted, which means one of 2 things. Either our understanding of evolution is fundamentally incomplete, or the RHD deletion is not what we think it is. Return to the Basque frequency, 47.2%, nearly half the population lacking the RHD gene. In 1975, a population geneticist named Cavalli Sforza published a comprehensive analysis of European blood group distributions. He called the Basque RN negative concentration an evolutionary enigma, not an anomaly, not a statistical outlier. An enigma, a puzzle whose solution contradicts the assumptions built into the question. Because here is what should happen under standard evolutionary theory. When a deleterious allele arises, one that reduces reproductive fitness, natural selection purges it. The strength of selection is proportional to the harm it causes and the frequency at which it appears. For Rho negativity, the harm is clear. Before medical intervention, hemolytic disease killed or damaged approximately 10% of RAF-incompatible pregnancies. That is a massive fitness cost. Frequency-dependent selection amplifies the purge. As RAS negative becomes rarer, the probability of RAS incompatibles matings increases. An RAS negative woman in a population where 99% of men are race positive has almost no chance of avoiding the incompatibility. Her lineage contracts. The mathematics predict extinction within 100 generations, and yet the Basques have maintained near 50% frequency for at least 1,000 generations, approximately 35,000 years, dating back to the Upper Paleolithic. Geneticists propose drift, founder effects, small population size amplifying random fluctuations, but drift is a neutral process. It does not favor alleles. It does not maintain balance. If the RHD deletion were truly neutral, if it had no effect beyond blood compatibility, then drift might explain its preservation in isolation. But it is not neutral. It kills children. Drift cannot maintain a lethal allele at 47% for 35,000 years. Something else is operating. In 2008, researchers in the Czech Republic began noticing patterns in psychological and behavioral data that had nothing to do with blood transfusions. They were studying the effects of latent Toxoplasma gondii infection, a parasite carried by approximately 30% of the global population, typically asymptomatic, transmitted through undercooked meat or contact with cat feces. Toxoplasma crosses the blood-brain barrier. It forms cysts in neural tissue, and it alters behavior. Infected rodents lose their fear of cats, increasing predation. An evolutionary strategy that returns the parasite to its definitive host. In humans, the effects are subtler: slower reaction times, increased risk-taking. In men, slight increases in testosterone. In women, shifts in personality traits. But the Czech researchers found something unexpected. The effects were not uniform across blood types. HERV-positive individuals, particularly heterozygotes, genotype DD, showed no measurable cognitive decline from Toxoplasma infection. HERV-negative individuals showed significant impairment, decreased psychomotor performance, prolonged reaction times. In a sample of military conscripts, infected Rh-negative drivers had a 2.4-fold higher risk of traffic accidents compared to uninfected Rh-negative drivers or infected Rh-positive drivers. The D antigen was somehow protective, not against infection. Negative and RR-positive individuals contracted Toxoplasma at similar rates, but against the neurological consequences. The researchers expanded their survey. HERV-negative men, regardless of Toxoplasma status, scored significantly worse on mental health-related variables. Higher self-reported anxiety, greater levels of psychological distress. Both Rh-negative men and women reported higher sexual activity and desire compared to Rh-positive peers, even after controlling for age, socioeconomic status, and relationship stability. Why would a blood protein, expressed on red blood cells, not neurons, affect anxiety? Affect libido? Affect whether you crash your car when infected by a parasite? Why would something that exists solely to carry oxygen through capillaries change how your brain processes threat? Unless the D antigen is not about blood. Unless it is about something deeper, metabolic, neurological, systemic, that we have not been measuring because we thought we were studying blood types. These were not small samples. These were population-level correlations replicated across multiple studies in different countries. Something about the presence or absence of the D antigen was affecting brain function. Then came 2020. The SARS-CoV-2 pandemic swept across populations, and epidemiologists began stratifying infection and mortality data by every available variable. Blood type emerged as a predictor. Nano-blood groups showed higher infection risk. And within each ABO category, RDR-positive individuals showed consistently elevated susceptibility compared to RDR-AR. The mechanism remains unclear. Hypotheses involve ACE2 receptor expression or differences in glycosylation patterns on cell surfaces, but the pattern was undeniable. RACE-CR-positive status, the presence of the D antigen, correlated with higher viral infection risk. RI-N-negative status correlated with resistance. Do you see the contradiction forming? For Toxoplasma, RS-positive is protective. For SARS-CoV-2, RES-negative is protective. For pregnancy, RS-positive avoids immune attack. For anxiety and mental health, RS-positive is associated with better outcomes. The D antigen is not simply good or bad. It is context-dependent. It is a trade-off. This is the signature of balancing selection. When two alleles persist in a population at stable frequencies for thousands of generations, evolution is not indifferent. It is not drifting. It is maintaining a polymorphism because both variants confer advantages under different circumstances. The textbook example is sickle cell trait in malaria endemic regions. Individuals with 2 copies of the sickle cell allele develop severe anemia and often die young. Individuals with 0 copies are fully susceptible to malaria. But heterozygotes, those with 1 normal allele and 1 sickle cell allele, gain resistance to malaria without suffering the full anemia. The polymorphism persists because the environment selects for genetic diversity. The raw system does function the same way. This is not hypothesis. This is pattern recognition. The 47.2% frequency in Basques is not an accident. It is an equilibrium maintained because RT-positive homozygotes, DD, heterozygotes, D, and RT-negative homozygotes, DD, each provide survival advantages under different threats. And the data do not hint. They declare dying heterozygotes may be the optimal genotype. Research on red blood cell antigen density revealed that ART-positive heterozygotes express approximately 17,720 D antigen sites per erythrocyte. RD-positive homozygotes express more, around 30,000 sites. RIS-negative individuals express 0. That middle number, 17,720, may represent a Goldilocks zone. Enough D antigen to provide protection against parasitic neuroinflammation. Not so much that it increases vulnerability to viral entry or immune overreaction. And the proposed mechanism involves membrane transport. The rough proteins are thought to facilitate ammonia, NH3, and carbon dioxide, CO2, transport across the red blood cell membrane. When pathogens like Toxoplasma trigger inflammation, or when hypoxia stresses the system, optimal gas exchange becomes critical. Too few Rs proteins, impaired transport, cognitive deficits. Too many, potential dysregulation, increased infection susceptibility. Heterozygotes, balanced. This would explain the Basque persistence. If the population faced sustained pathogenic pressure, parasites, environmental toxins, altitude hypoxia in mountain valleys, then natural selection would favor the maintenance of both alleles. DD individuals would outcompete DD and DD under those specific conditions. Some Rs-negative infants would die from hemolytic disease. But the overall fitness of the population, its resistance to the dominant environmental threats, would increase. The cost of infant mortality would be offset by the advantage of infection resistance. We have just crossed a threshold. Hour 1 asked, why does the deletion exist? Hour 2 asks, what was it defending against? And that question opens a door we may not want to enter. Because of heterozygote advantage explains the persistence, it does not explain the frequency. Why 47% in Basques when neighboring populations show 15%? What localized threat, what environmental pressure unique to the Franco-Cantabrian region was so deadly that evolution drove the polymorphism to near 50-50? What leaves that kind of genetic scar? The paradigm has shifted. The RHD deletion is not a flaw. It is not a remnant of ancient aliens or lost civilizations. It is an adaptation. And the fact that we cannot name what it was adapting to does not diminish the certainty of the signal written in Basque blood. Something in Paleolithic Europe was deadly enough that mothers' bodies were willing to sacrifice their own children generation after generation for 35,000 years to maintain genetic diversity. What kind of threat demands that price? The RHD deletion is not a flaw. It is not a remnant of ancient aliens or lost species. It is an adaptation. And the fact that we do not yet know what it was adapting to does not diminish the certainty of the signal. Something in the environment of Paleolithic Europe, something persistent, something deadly, selected for a genetic diversity so extreme that mothers' bodies were willing to sacrifice their own children to maintain it. We must now ask, what kind of threat justifies that cost? RIA-negative men, higher anxiety, elevated stress hormones. RIV-negative women, higher sexual desire, more frequent mating. RA-negative drivers infected with Toxoplasma, 2.4 times more likely to crash. RIFist of individuals, higher SARS-CoV-2 infection rates. This is not a blood type. This is a phenotype that reaches from red blood cells into the brain, into behavior, into evolutionary fitness, in ways that transfusion medicine never predicted. Before we can explain what the RAS system is protecting against, we must catalog what it is doing. Let us begin with a hypothesis that sounds like folklore, but is grounded in immunology. It is called the grandmother theory. Consider an RS-negative woman giving birth to her first child, an RS-positive daughter. During delivery, fetal blood crosses into the mother's circulation. Anti-D antibodies begin forming, but slowly. The daughter is born healthy. Now fast forward 25 years. That daughter, now RS positive herself, becomes pregnant with her first child. Your child, if you are following this lineage forward. Standard medical teaching says the daughter's first pregnancy should be safe. She has never been pregnant before. She has no pre-existing anti-D antibodies. But the grandmother theory asks, what if she was sensitized at birth? If sufficient RA-positive maternal blood entered the daughter's circulation during her own delivery, if her RF-negative grandmother's blood cells, still circulating in the newborn infant's body during those first hours, then the daughter's infant immune system might have encountered the D antigen while it was still developing tolerance. Her immune system would classify D antigen as foreign from the very beginning, not through infection, not through transfusion, through birth itself. And decades later, when she carries an RORS-positive fetus, her memory B cells, trained in infancy, would activate immediately. Her first pregnancy would fail. This hypothesis has never been definitively proven. It exists at the edge of immunological plausibility, but it suggests something more disturbing than standard R incompatibility, that the conflict is not merely between mother and child, but across 3 generations. That the immune system remembers traumas from birth, encoded in cellular memory, waiting decades to express. That Rh negativity might propagate not through genetic advantage, but through a cascade of immunological conditioning where each generation's exposure shapes the next generation's response. A hereditary immune imprinting. Now, shift to psychology. The Czech studies that identified Toxoplasma susceptibility also measured baseline psychological profiles independent of infection. RF-negative men reported higher anxiety scores, greater neuroticism, elevated stress responses even in controlled laboratory settings. Rh-negative women showed similar trends, though less pronounced. The effect size was small. Psychology is multifactorial, shaped by genetics, environment, culture, but it was statistically significant across thousands of participants. Why would a blood protein affect mood? The ROT proteins are expressed almost exclusively on red blood cells and, to a lesser extent, in renal tubules. They are not diurnal. They do not cross the blood-brain barrier in significant quantities, but they affect what crosses. If ART proteins regulate gas exchange, ammonia and carbon dioxide efflux from red blood cells, then their absence might subtly alter the chemistry of blood perfusing the brain. Elevated ammonia can cause irritability, cognitive fog, anxiety. Impaired CO2 clearance can shift blood pH, affecting neurotransmitter receptor sensitivity. Small effects, chronic effects. The kind of effects that would not prevent survival, but would shape personality over a lifetime. And then there is the sex data. Multiple independent studies in the Czech Republic, in Turkey, in the United States found that RES-negative individuals, both male and female, report higher sexual activity and stronger sexual desire compared to RES-positive controls. The correlation persists even when adjusting for age, relationship status, and cultural background. No one knows why. Evolutionary psychologists have speculated that if RAN negativity carries a reproductive cost, if RAN-negative women lose children to hemolytic disease, then selection might favor behavioral compensation, higher libido, more frequent mating, more pregnancies to offset the losses. But this is post hoc reasoning. It assumes that behavior evolves to counteract genetic penalties, which is not how natural selection operates. Selection acts on outcomes, not intentions. If RS-negative women are having more children overall, despite hemolytic disease losses, then the deletion is not deleterious. It is advantageous. And if sexual behavior is genuinely affected by Rh status, then the mechanism must be physiological, not psychological. Hormones. Neurotransmitters, something biochemical, return to Toxoplasma. The 2.4-fold increase in traffic accidents among infected RAS-negative drivers is not subtle. That is a massive effect for a supposedly asymptomatic infection. But here is what the data also showed. RA-positive individuals, specifically heterozygotes DED, showed no increase in accident risk even when infected. They carried the parasite. Brain cysts were present, but reaction times remained normal. The D antigen was somehow shielding neural function from parasitic interference. The proposed mechanism involves neuroinflammation. Toxoplasma cysts in the brain do not directly destroy neurons. They trigger low-grade chronic inflammation. Microglia activate. Cytokines increase. Dopamine metabolism shifts. In animal models, this inflammation impairs neurotransmission, particularly in pathways involving motor coordination and risk assessment. Exactly the functions compromised in infected Rh-negative drivers. But if red blood cells carrying optimal D antigen density can buffer ammonia and CO2 more efficiently, then they might mitigate the hypoxic and metabolic stress caused by neuroinflammation. The brain suffers less. Reaction times stay sharp. The heterozygotes, with their 17,720 antigen sites per cell, sit in the optimal range. DD homozygotes with 30,000 plus sites overcorrect. Too much membrane activity, potential dysregulation. D homozygotes with 0 sites cannot compensate at all. The protection is not about having the D antigen. It is about having the right amount of D antigen. This is why the SARS-CoV-2 data initially seemed contradictory. Rho-positive individuals showed higher infection rates. Rha-negative individuals showed relative resistance. But infection susceptibility and neurological resilience are different selective pressures. A virus that enters through ACE2 receptors in the respiratory tract does not care about ammonia transport across red blood cell membranes. But a parasite that colonizes the brain and disrupts metabolic homeostasis does. The RAS system is not optimized for one threat. It is balancing multiple threats simultaneously. Now consider the geographic distribution again, but with this framework. Why do Basques show 47% RA's negative frequency, while neighboring Spanish populations show 25% and Middle Eastern populations show less than 10%? If heterozygote advantage is real, then the optimal frequency of the deletion depends on the strength and type of selective pressure. In malaria-endemic Africa, sickle cell heterozygotes reach 10 to 20% frequency in some regions, just enough to provide population-level resistance without too many homozygous deaths. In temperate Europe, where malaria is absent, sickle cell is nearly extinct. Different environments. Different equilibria. So what was endemic to the Basque Country that was not endemic to, say, the Po Valley in Italy or the Rhine corridor in Germany? What localized selective pressure could push RAS-negative frequency to 47%, double or triple the European average, and maintain it for 35,000 years? Toxoplasma is global. It does not explain regional differences. Altitude. The Basque Country includes coastal lowlands. Hypoxia is not a universal Basque experience. Dietary toxins. Possibly, but the region's traditional diet, fish, dairy, legumes, is not unique. Inbreeding? The Basques were isolated, yes, but so were the Sardinians, the Icelanders, the Sami. None show RS-negative frequencies approaching 47%. Here is where the anomalies sharpen into genuine mystery. Because the Basque language, Euskara, is not simply old, it is unrelated. Linguists have compared it to every language family in Eurasia. No connections. Some surface similarities to Caucasian languages, Chechen, Georgian, but no deep structural alignment. It is as if Euskara emerged independently or arrived from a lineage now extinct. and the Basque genome shows similar isolation. Y chromosome haplogroup R1b reaches extreme frequencies. Mitochondrial haplogroup H, likewise. These are markers of the first Paleolithic Europeans, Cro-Magnon hunter-gatherers who entered the continent 45,000 years ago, before the Neolithic farmers arrived from Anatolia. The Basques are, genetically, a relic population. But relics should not thrive. Isolated populations accumulate deleterious mutations through genetic drift. They lose diversity. They become fragile. The Basques did the opposite. They maintained a polymorphism so balanced, so finely tuned, that it has persisted for 1,000 generations without collapsing into fixation or extinction. That does not happen by accident. In the 1970s, frustrated researchers proposed alien intervention. We will not dignify that hypothesis. It replaces one mystery with an unfalsifiable claim, explains nothing, and wastes the pattern embedded in the data. But we understand the impulse. When every conventional model fails, the mind reaches for the impossible. We will not make that error. But we must acknowledge what the aliens of the gaps argument gets right. The Ross system feels like a species barrier. The immune violence, the geographic clustering, the psychological and behavioral correlations. It feels like two lineages colliding inside one genome, not because aliens intervened, but because evolution itself is a collision of genes, of environments, of pathogens that rewrite us, and adaptations that rewrite them back. The failure of genetic drift as an explanation is now complete. Drift cannot maintain 47% frequency of a lethal allele for 35,000 years in a small population. The failure of simple mutation as an explanation is also complete. The RHD deletion is not random noise. It correlates with too many phenotypes: anxiety, libido, infection response, neurological resilience, to be neutral. What remains is the radical hypothesis. Heterozygote advantage. Balancing selection. A genetic polymorphism maintained because both variants provide survival benefits under different conditions, and the optimal genotype is the combination of the two. But if that is true, then the next question is unavoidable. What was the Basque environment selecting for? What threat was so persistent, so deadly, so localized to the Franco-Cantabrian region that it required a genetic diversity extreme enough to tolerate the death of children? And why, if this threat existed 35,000 years ago, do we see no trace of it in the archaeological record? No mass graves, no evidence of epidemic disease, no environmental catastrophe. Only the blood, only the deletion, only the silence of a language that remembers no origin. We have catalogued the anomalies. We have rejected the aliens. We have dismantled genetic drift. What remains is the radical hypothesis, the one grounded not in speculation, but in evolutionary mechanics documented in every system where balancing selection operates. The question evolution has been encoding into Basque genomes for 35,000 years. What is the D antigen actually for? We have catalogued the anomalies. We have dismantled the old theories. Now we must construct the new one, not by invoking the supernatural, but by examining the mechanics of the D antigen itself, its structure, its density, its biochemical function, and asking, what would it take for evolution to decide that this protein, at this precise concentration, was worth killing for? There is a principle in evolutionary biology that sounds paradoxical until you understand it. Sometimes the way to preserve a population is to preserve its contradictions, not to eliminate variation, not to select for a single optimal genotype, but to maintain genetic diversity, even at the cost of individual deaths, because diversity itself becomes the adaptation. This is called balancing selection, and it may be the only mechanism that explains the Basque blood. Let us return to the textbook example. Sickle cell trait. In sub-Saharan Africa, in regions where Plasmodium falciparum malaria is endemic. A specific mutation in the beta-globin gene produces abnormal hemoglobin. Individuals who inherit 2 copies of the sickle cell allele, genotype SS, develop sickle cell disease. Their red blood cells deform into rigid crescents under low oxygen conditions. Cells rupture. Organs fail. Most do not survive to adulthood. Individuals with 0 copies, genotype AA, have normal hemoglobin, but they are fully susceptible to malaria. In high transmission areas, childhood mortality from malaria can exceed 20%. Heterozygotes, genotype AS, carry 1 normal allele and 1 sickle cell allele. They produce enough normal hemoglobin to avoid severe anemia, and the presence of abnormal hemoglobin disrupts the malaria parasite's life cycle inside red blood cells, providing substantial resistance to infection. This is heterozygote advantage. The heterozygous genotype has higher fitness than either homozygous genotype. Natural selection cannot eliminate the sickle cell allele because doing so would eliminate malaria resistance. It cannot fix the sickle cell allele because doing so would cause universal anemia. The population reaches an equilibrium, typically 10 to 20% sickle cell allele frequency, where the benefits of malaria resistance balance the costs of anemia deaths. The polymorphism is maintained not despite the deaths, but because of them. Evolution is not gentle. It calculates in populations, not individuals. Now apply this framework to the Ross system. 3 genotypes. DD homozygotes, 2 functional RHD genes. High D antigen expression, approximately 30,000 sites per red blood cell. D heterozygotes, 1 RHD gene. Moderate D antigen expression, approximately 17,720 sites per cell. D homozygotes, 0 RHD genes. No D antigen. RAF, negative. If heterozygote advantage operates here, then DD individuals must have higher overall fitness than DD or DD under the selective pressures present in the Basque environment, and the data suggest they do. The key is not the presence or absence of the D antigen, but its density. Red blood cell membranes are not static barriers. They are active interfaces, studded with proteins that regulate what enters and exits. The RAF proteins, encoded by RHD and RHCE, are thought to function as gas channels, facilitating the transport of ammonia and carbon dioxide across the membrane. This is not trivial housekeeping. This is survival. Ammonia is a metabolic waste product, toxic at elevated concentrations. It is produced continuously by cells throughout the body, muscle tissue, gut bacteria, protein catabolism in the liver. Red blood cells must clear ammonia from tissues and transport it to the liver and kidneys for conversion into urea. If RO protein density is too low, ammonia clearance is impaired. Tissues accumulate the toxin. The brain, exquisitely sensitive to metabolic disturbances, suffers first. Irritability, cognitive fog, anxiety. The exact psychological profile observed in Rh-negative individuals. Carbon dioxide follows a similar logic. Cellular respiration produces CO2. Red blood cells bind it, buffer it, transport it to the lungs for exhalation. Efficient CO2 transport maintains blood pH within the narrow range required for enzyme function, neurotransmitter signaling, and cellular homeostasis. If transport is sluggish, blood becomes acidic. Respiratory acidosis. Fatigue. Impaired cognition. Again, the symptoms correlate with RES-negative status, particularly under metabolic stress. But here is the crucial insight. More R protein is not always better. DD homozygotes, with 30,000 plus D antigen sites, may overexpress membrane transport activity. Too much gas flux. Dysregulation of intracellular ion balance. Under certain conditions, viral infection, for example, this might make cells more susceptible to pathogen entry or immune-mediated lysis. The SARS-CoV-2 data, where R-positive individuals showed higher infection rates, hint at this vulnerability. D. Heterozygotes occupy the Goldilocks zone, 17,720 sites per cell, enough to efficiently clear ammonia and CO2 under normal conditions, enough to buffer metabolic stress during infection or inflammation, specifically the neuroinflammation caused by parasites like Toxoplasma gondii. Not so much that membrane activity becomes a liability during viral challenge. The heterozygote is resilient across a range of threats. The homozygotes are each optimized for narrower conditions. This is the mechanism by which balancing selection maintains polymorphism. If the environment alternates between pressures, parasitic infection one generation, viral epidemic the next, nutritional stress during famines, hypoxic stress at altitude, then no single genotype dominates. DD individuals thrive during parasite outbreaks. D individuals thrive during viral pandemics. D individuals survive both, though perhaps not as spectacularly as the specialists. Over millennia, the allele frequencies stabilize at a ratio that maximizes population-level survival. But why 47% in Basques? Why not 15% like the rest of Europe? Because the selective pressure in the Basque Country was more intense. Consider what 47% RS-negative frequency means in terms of genotype distribution. If we assume Hardy-Weinberg equilibrium, a population where alleles mix randomly, then the frequency of the deletion allele, D, is approximately 0.69. The frequency of the functional RHD allele, D, is approximately 0.31. This predicts DD homozygotes, 9.6%, D heterozygotes, 43%, D homozygotes, 47.4%. Nearly half the population is heterozygous. This is the signature of strong balancing selection, pushing toward a 1-1-1 ratio of alleles. Compared to sickle cell regions, where the mutation allele typically reaches 10 to 20%, producing roughly 20 to 40% heterozygotes. The Basque ratio is double. That implies the fitness advantage of DD heterozygotes, relative to the homozygotes, was substantially greater than the advantage seen in sickle cell trait. Or, and this is darker, it implies that both homozygous genotypes carried significant fitness costs under Basque conditions, making the heterozygote the only reliably viable option. What could cause that? What selective regime punishes both extremes equally? A parasite that targets high RER protein expression and a separate pressure, viral, environmental, dietary, that punishes low expression. Or a single complex pathogen with multiple invasion strategies, where DD and DD individuals are each vulnerable to different stages of infection, but DD individuals can resist the full life cycle. The archaeological record provides hints but no definitive answers. The Basque Country during the Upper Paleolithic was densely populated by Cro-Magnon groups. The caves, Altamira, Icain, Santa Mamigne, contained evidence of sophisticated hunter-gatherer societies. They hunted red deer, ibex, wild boar. They fished Atlantic salmon. They gathered shellfish along the coast, and they lived in close proximity to animals. Zoonotic diseases, pathogens that jump from animals to humans, are a constant pressure in such environments. Toxoplasma is zoonotic, transmitted by cats but also by undercooked game meat. So are various helminths, parasitic worms that colonize the gut and sometimes the brain. So are vector-borne diseases carried by ticks and fleas in temperate forests. It is plausible that the Basques faced a pathogen load distinct from neighboring regions, perhaps due to unique ecological conditions in the Pyrenean valleys or cultural practices around animal husbandry and food preparation. But plausible is not proof. The pathogens themselves left no fossils. We infer their existence from the genetic scars they carved into the survivors. There is another, more speculative possibility: the research on age-related physiological decline. Some studies suggest that R-positive individuals, particularly heterozygotes, show slower rates of cognitive decline in old age compared to Rh-negative individuals. If Rh protein density affects long-term neural health, then populations with longer lifespans or where elder knowledge was critical for survival might experience selection for genotypes that preserve cognition. The Basques have historically shown longevity. Even in pre-modern populations, living to 60 or 70 was not uncommon in the mountains, where diet and lifestyle differed from agricultural lowlands. Perhaps the heterozygote advantage is not merely about resisting infection. Perhaps it is about aging well. This expands the time horizon of selection. Malaria kills children. Selection against sickle cell disease is swift and visible. But neurological resilience across decades? That selects quietly, invisibly, shaping allele frequencies through the accumulated survival of individuals who remained cognitively sharp enough to hunt, to teach, to lead. The advantage compounds over a lifetime. And in a small, isolated population, that compounding effect might be sufficient to drive the deletion allele to 47%. But we must also confront the cost: hemolytic disease of the newborn. In a population where 47% are DD and 43% are DD, the vast majority of pregnancies involve at least one RDH-negative parent. When an Rh-negative woman DAT mates with a heterozygous man, D, 50% of her children will be Rh-positive. When she mates with an Rho-positive homozygous man, DD, 100% of her children will be positive. Given the frequency distributions, roughly 30 to 40% of all pregnancies in the Basque population would be Rho incompatible. Before Rogam, that meant second and third pregnancies carried a 10 to 15% risk of severe hemolytic disease. Thousands of infants over the generations. Dead. Brain damaged. Stillborn. This is the price of polymorphism. And evolution paid it willingly because the alternative, losing the heterozygote advantage, allowing the population to collapse into DD or DD homozygosity, was worse. The math is brutal, but it is consistent. If DOT heterozygotes have even a 10% fitness advantage over homozygotes, through infection resistance, cognitive resilience, metabolic efficiency, then the loss of 5 to 10% of DD pregnancies to hemolytic disease is acceptable at the population level. The polymorphism stabilizes. The deaths continue, but the population thrives. This is not speculation. This is how balancing selection operates in every system where it has been rigorously studied. In MHC diversity, where immune recognition genes are maintained at extreme polymorphism despite individual incompatibilities. In ABO blood groups, where O, A, and B alleles persist because each confers resistance to different pathogens. In lactase persistence, where the ability to digest milk in adulthood is beneficial in pastoralist societies but costly elsewhere. Evolution does not design perfect organisms. It designs populations that can survive unpredictable environments by containing multiple solutions, even when those solutions conflict. The Basques are not anomalous. They are exemplary. They demonstrate balancing selection pushed to an extreme, where the selective pressure was so sustained, so multifaceted, that the population converged on a near 1 allele ratio and maintained it for 35,000 years. The RHD deletion is not a mutation that should have disappeared. It is half of a two-part adaptation, inseparable from the presence of the D antigen itself. Remove either allele and the system collapses. But this raises a final, haunting question. If the heterozygote advantage was powerful enough to maintain 47% RS-negative frequency in the Basques, why did it not spread? Why didn't neighboring populations, the Spanish in Castile, the French in Aquitaine, converge on the same equilibrium? The Basques were not perfectly isolated. There was gene flow, intermarriage, migration, And yet the deletion frequency drops sharply outside the Franco-Cantabrian core. Why? Two possibilities. First, the selective pressure was geographically localized. Whatever pathogen or environmental stressor drove the polymorphism existed only in the Basque valleys. Outside those valleys, the advantage vanished, and genetic drift or weak selection against hemolytic disease reduced the deletion frequency. Second, the cultural and linguistic isolation of the Basques was so profound that gene flow was negligible. The deletion did not spread because the Basques did not interbreed with outsiders at rates sufficient to homogenize allele frequencies. Both may be partially true. What remains undeniable is this: the Basques carry in their veins a genetic equilibrium sculpted by forces we can infer but not fully name, a pathogen we cannot identify, a pressure that left no trace except the blood itself, and the fact that their language, Chuskara, remains unrelated to any other language on Earth, suggests that this isolation, this genetic and cultural distinctiveness, may reach back to a time before recorded history, to the Paleolithic, to the first humans, to a lineage that adapted to something the rest of Europe never encountered, or survived without needing to adapt so drastically. The radical theory is not radical at all. It is simply evolutionary biology, applied rigorously to an extraordinary case. The raw polymorphism is maintained by heterozygote advantage. The advantage involves metabolic resilience, ammonia and carbon dioxide transport optimized at a specific D-antigen density. The cost is hemolytic disease, which evolution tolerates because the population-level benefit exceeds the individual-level deaths. The mystery is not how it persists. The mystery is why the selective pressure was so much stronger in one isolated population than anywhere else on Earth. And that mystery, that localized, invisible force that shaped the Basque genome, opens the door to something larger. Because if balancing selection can maintain a lethal polymorphism at 47% frequency, what else might it have shaped? What other human traits, psychological, physiological, behavioral, are not fixed adaptations but balanced contradictions, preserved because diversity itself is the strategy? We must now ask, what does it mean to be human if humanity is not one optimized design, but a collection of competing, incompatible strategies held in precarious balance? We have been telling a story about blood, about a deletion in a single gene, about mothers and children, about a population isolated in mountain valleys preserving a genetic contradiction that should not exist. But the story is not about blood. It is about what it means to be alive in a body assembled from compromises, where every adaptation is a trade-off, every strength contains a vulnerability, and survival depends not on perfection, but on the capacity to balance opposing truths. The raw system is a window, and through that window we glimpse something far stranger than blood incompatibility. We glimpse the architecture of the human genome itself and the ancient negotiations that made us possible. Let us return to the 8%, the viral code threaded through your DNA. The 203,000 copies of human endogenous retroviruses inscribed into your chromosomes by infections that occurred millions of years before you were born. We called them fossils, genetic ruins, but that metaphor is incomplete, because some of these viruses are not inert, they are functional. Consider the genes that regulate your digestion. The amylase genes encoding enzymes that break down starch are flanked by HERV sequences that act as enhancers, regulatory switches. When those viral elements are active, amylase production increases. You can digest bread, rice, potatoes more efficiently because a retrovirus long ago inserted itself near a metabolic gene and now modulates its expression. The virus became part of the machinery. Or consider hemoglobin, the protein that carries oxygen in your blood, the molecule at the center of the RA story. The beta-globin gene cluster on chromosome 11 is surrounded by HERV-derived regulatory elements. These elements fine-tune the timing and intensity of hemoglobin production during fetal development. Without them, the transition from fetal to adult hemoglobin, critical for survival at birth, would be impaired. A virus rewrote the instructions for how you breathe. This is the first cosmic implication. You are not a purely human design. You are a chimera, a genetic collaboration between an ancestral primate genome and the parasites that infected it. The line between you and invader does not exist. It dissolved millions of years ago in the germ cells of a mammal whose name we will never know, when a retrovirus integrated into a chromosome and, through sheer accident, improved the fitness of its host. But here is the terror nestled inside the wonder. If HERVs can enhance essential genes, they can also destroy them. Most HERV sequences are silent, suppressed by epigenetic modifications, chemical tags that lock the DNA into an inactive state. Methylation, histone deacetylation, the cell's defense against its own viral ghosts. But suppression is not permanent. Inflammation can reactivate HERVs. So can environmental toxins. So can other infections, acting as triggers. When a HERV wakes, it begins producing proteins, viral envelope proteins, reverse transcriptase, structural components, and the immune system reacts. Sometimes the reaction is precise. The infected cell is eliminated, the threat contained. Other times, the reaction is catastrophic. Multiple sclerosis, a neurodegenerative disease where the immune system attacks the myelin sheath insulating nerve fibers in the brain and spinal cord. For decades, the cause was unknown. Genetic susceptibility, yes. Environmental triggers, perhaps. But no single pathogen could be identified. Then, in the 1990s, researchers discovered elevated levels of HERV-W envelope protein in the cerebrospinal fluid and brain lesions of MS patients. The protein, normally silent, was being expressed, and the immune system, recognizing it as foreign, was launching an inflammatory assault that inadvertently destroyed myelin. The virus was not an external infection. It was already there, written into the genome, waiting. And when conditions aligned, perhaps an initial viral infection, perhaps chronic stress, perhaps a specific combination of genetic and environmental factors, it activated. The body began attacking itself because it could not distinguish the endogenous retrovirus from the neural tissue surrounding it. Schizophrenia follows a similar pattern. The disease typically manifests in late adolescence, precisely when the brain undergoes massive synaptic pruning, the elimination of excess neural connections to optimize cognitive efficiency. HERV expression in the prefrontal cortex spikes during this developmental window. In some individuals, the viral proteins dysregulate the pruning process. Too many synapses are eliminated. Neural circuits fragment. Auditory hallucinations. Disorganized thought. The subjective experience of reality unraveling, not because of a new infection, but because of an ancient one reactivated at the worst possible moment. And cancer. Some HERVs encode proteins that, when expressed in the wrong cell type, promote uncontrolled growth. They mimic growth factors. They inhibit tumor suppressors. Certain leukemias and lymphomas show aberrant HERV activation in malignant cells. The virus does not cause the cancer directly. Cancer is multifactorial, requiring mutations in multiple genes, but it contributes. It tips the balance toward malignancy. A genetic joker, flipping from benign to pathogenic, depending on context. This is the second cosmic implication. The greatest threats to your health may not come from outside. They come from inside. From the viral code you inherited, dormant but never truly erased. Capable of reactivation under conditions that evolution could not prepare you for. Because evolution did not anticipate modern lifespans. It did not anticipate industrial toxins, chronic psychological stress, autoimmune triggering vaccines, or the epigenetic disruptions caused by processed diets. The HERVs were stable in Paleolithic environments. They are not stable now. But before we succumb to despair, we must confront the other side of the equation. The reason HERVs exist at all, despite the risks. The reason they were not purged by selection. Because some of them are not merely tolerated. They are essential. You exist, this must be said plainly, because approximately 25 million years ago, an ancestor of all placental mammals was infected by a retrovirus. The virus integrated into the germline. It passed to offspring. And one of its genes, the invade gene encoding the viral envelope protein, was repurposed. The protein, now called syncytin, allows trophoblast cells in the developing placenta to fuse together, forming a multinucleated layer, the syncytiotrophoblast, that mediates nutrient and gas exchange between mother and fetus. Without syncytin, there is no functional placenta. Without a placenta, there is no gestation beyond a primitive yolk sac. Mammals would still lay eggs, like reptiles, or carry embryos in pouches, like marsupials. But the prolonged intrauterine development that defines placental mammals, elephants gestating for 22 months, humans for 9, would be impossible. The intimacy between mother and fetus, the direct vascular connection, the ability to nourish a large-brained offspring to full term inside the body, all of it depends on a domesticated virus. This is not metaphor. The syncytin gene still shows the structural hallmarks of a retroviral envelope protein. It has the fusion peptide. It has the transmembrane domain. If you isolated syncytin and expressed it in a cell culture, it could still mediate membrane fusion, exactly as it did when it was part of an active virus. But evolution captured it, tamed it, integrated it so deeply into mammalian development that its viral origin is now irrelevant. The invader became indispensable. Think about what this means for the R story. The placenta, the organ where R incompatibility becomes lethal, is itself a viral invention. The immune war between RE-negative mothers and RE-positive fetuses unfolds across a structure built by ancient retroviruses. It is a conflict mediated by viral domestication, where the outcome depends on the balance of surface proteins that may themselves have been sculpted by viral host coevolution. We are not merely infected by our past. We are composed of it. This is the third cosmic implication. Human reproduction, the act of creating new life, is an improvisation built on hijacked viral machinery. You were not born from a purely human blueprint. You were born from a negotiation between your mother's genome and the ancient viruses embedded within it, executing a developmental program that would not exist if not for parasitic code repurposed as architecture. And yet the system is fragile. Syncytin expression must be tightly regulated. Too little, and the placenta fails to develop, implantation disorders, miscarriage. Too much, and abnormal trophoblast invasion occurs, preeclampsia, a hypertensive disorder that can kill both mother and child. The virus, even domesticated, retains the capacity to malfunction. Some researchers now suspect that preeclampsia may correlate with dysregulated HERV expression, not just syncytin, but other retroviral elements activating inappropriately during pregnancy. The immune system becomes confused. It cannot distinguish placental tissue from viral threat. Inflammation surges, blood pressure spikes, and we return, once again, to the maternal-fetal interface as a battleground, where the line between self and other, between nurture and attack, is determined by molecular signals inherited from viruses that infected our ancestors before mammals existed in their modern form. Now, pull the lens back further. Consider the evolutionary time scale. The oldest HERVs integrated 100 million years ago. The youngest are integrating now, polymorphic insertions unique to specific human lineages, still spreading through populations. Viral evolution is not a chapter that closed in the Paleolithic. It is ongoing. Every generation, Endogenous retroviruses have the potential to move, to copy themselves to new locations in the genome, to insert near genes and alter their regulation. Most insertions are neutral or deleterious. They disrupt function. They are eliminated by selection. But occasionally, an insertion lands in a regulatory region and improves fitness. A new enhancer, a new splice site, a new gene born from viral sequence. Evolution is still writing. This means that human genetic diversity, the variation between individuals, between populations, is not just the product of point mutations accumulating slowly over millennia. It is also the product of retrotransposition, viral elements jumping through the genome, creating structural variants, deleting genes, duplicating genes, rewiring regulatory networks. The RHD deletion itself may have originated through such a mechanism. A retroviral insertion followed by recombination that excised the entire gene. We do not know, but the possibility is there. In the architecture of chromosome 1, where HERV sequences flank the RS locus like molecular scars. The fourth cosmic implication: you are not a stable entity. Your genome is a dynamic text, still being edited by parasitic elements that existed long before you and will persist long after. The illusion of the human genome as a single definitive sequence is precisely that, an illusion. There is no one human genome. There are 8 billion variations, each carrying a unique constellation of viral insertions, deletions, and polymorphisms. And buried within that variation are the seeds of future adaptations, future diseases, future species. Because if HERVs can maintain polymorphisms like the R system, where lethal incompatibilities persist because the diversity itself confers advantage, then speciation itself may be a viral process, not in the science fiction sense of alien DNA, but in the rigorous evolutionary sense. Populations diverge not just through geographic isolation and drift, but through differential viral integration events that create genetic incompatibilities between lineages. Imagine two populations separated for 50,000 years. Each experiences different retroviral insertions, different HERV activations, different regulatory rewiring. When they meet again and interbreed, their offspring may carry incompatible gene networks, viral enhancers from one lineage activating genes that should be silent or suppressing genes that should be active. The hybrid genome becomes a battlefield, and selection favors individuals who can tolerate the contradictions. Heterozygotes, like the DD genotype in the Ross system, who balance the incompatibilities and survive. This is speculative. This is at the frontier of evolutionary genomics, but it is grounded in observed phenomena, hybrid incompatibility in closely related species, where specific gene combinations cause sterility or developmental failure. The Ross system is a mild version of this, not full speciation, but a glimpse of the forces that could, over longer timescales, fragment a single species into reproductively isolated lineages. The Basques, with their 47% Rh negative frequency, their linguistic isolation, their genetic distinctiveness, may represent an early stage of such divergence. Not a separate species, but a population whose genome has been sculpted by selective pressures sufficiently unique that they carry genetic signatures found nowhere else on Earth at comparable frequency. They are a living fossil, not of an ancient human subspecies, but of a specific evolutionary trajectory, a path that diverged 35,000 years ago and has been maintained. Against the statistical improbability of drift and migration, through sheer adaptive value. And here is the haunting possibility. If we eradicate the selective pressures, if modern medicine prevents hemolytic disease, if antiparasitic drugs eliminate toxoplasma, if sanitation reduces pathogen load, then the raw polymorphism will no longer be maintained. The deletion allele will decline, slowly, over generations, the Basque genome will homogenize with the rest of Europe. The heterozygote advantage will vanish because the threats it defended against no longer exist. We will have erased, through medical progress, a 35,000-year-old adaptation. Is this victory or loss? Is it ethical to preserve genetic diversity for its own sake, even if it means tolerating hemolytic disease in the absence of ROGAM? Or is it acceptable to let selection relax, to allow the polymorphism to collapse, knowing that we are closing an evolutionary chapter that may never reopen? These are not abstract questions. They are the questions every human society will face as we gain the capacity to edit our own genomes, to silence HERVs, to eliminate deleterious alleles before birth. Do we preserve our viral heritage because it might confer advantages we do not yet understand? Or do we delete it, accepting the risk that we are removing adaptations calibrated for futures we cannot predict? The cosmos is indifferent. Evolution has no preference, but we must choose, and the choice we make will determine not just the genetic future of humanity, but the philosophical one. Are we the curators of our inherited contradictions, or the architects of a new design? The future is already being written in clinical trial registries and gene therapy labs. Not the distant future of science fiction, starships, and uploaded consciousness. The immediate future, the next 2 decades, where the questions we have been circling: what are HERVs doing? Should we silence them? Can we rewrite the viral heritage embedded in our genome? Transition from philosophical speculation to medical intervention. The age of passive observation is ending. The age of editing has begun. In 2019, a Swiss pharmaceutical company began phase 2 clinical trials for a drug called temolizumab. It is a monoclonal antibody, a precisely engineered immune protein designed to bind to and neutralize a single target, the HERV envelope protein, the same viral protein implicated in multiple sclerosis, the same protein that, when activated, triggers the inflammatory cascade that destroys myelin and fragments neural circuitry. Temolizumab does not attack the virus directly. You cannot attack something already written into your chromosomes. Instead, it neutralizes the protein the virus produces, preventing it from interacting with immune cells and initiating the autoimmune response. Early results showed promise. Patients with relapsing remitting MS experienced reduced disease progression. Inflammatory markers decreased. MRI scans showed fewer new lesions forming in the brain. The drug was not a cure. MS is multifactorial: genetic susceptibility, environmental triggers, cumulative damage over years. But temolizumab represented a new paradigm, treating disease by managing our viral ancestry rather than pretending it does not exist. The company expanded trials to include type 1 diabetes, another autoimmune condition linked to HERV activation. In type 1 diabetes, the immune system attacks insulin-producing beta cells in the pancreas. The disease typically manifests in childhood and requires lifelong insulin therapy. Recent evidence suggests HERV proteins may be expressed in pancreatic tissue during viral infections, triggering an autoimmune response that persists long after the initial infection clears. If temolizumab can block HERV during the critical window, when beta cells are under attack, it might prevent or delay disease onset, not by curing diabetes, by interrupting the viral contribution to its development. This is the near-term scenario: targeted therapies that acknowledge HERVs as contributors to disease and attempt to modulate their effects without eradicating them entirely. It is conservative, incremental. It works within the constraints of the genome we inherited, but it is not the only path forward. CRISPR-Cas9 gene editing has made it theoretically possible to delete HERV sequences from human chromosomes. Not in living adults. The logistical challenges of editing trillions of somatic cells are immense, but in embryos, in germline cells, in the generation not yet born, a single embryo edited before implantation could have specific HERV families silenced. Picture this: a child born in 2035. Before implantation, their embryo was screened, edited, optimized, HERV-W silenced, no multiple sclerosis pathway, HERV-K locked, no cancer contribution from viral reactivation, RHD deletion corrected, no hemolytic disease risk. They grow up healthy, perfect, the poster child for precision genomics. Then, in 2060, a novel zoonotic pathogen emerges from thawing permafrost. A parasite similar to Toxoplasma but more aggressive. It targets the very HERV sequences that were silenced, uses them as entry points, and when it finds them locked, it cannot complete its life cycle. The edited children, the optimized generation, have no defense against the virus that follows the parasite. They die at rates of 40%. The unedited children, the ones carrying their flawed genomes with active HERVs, survive at rates of 90%. This is not science fiction. This is the gamble. Not removed, that would require excising 8% of the genome, an impossibility, but epigenetically locked, methylated, made permanently inactive. The child born from that embryo would still carry the viral sequences, but they would never activate. No HERV-W proteins, no multiple sclerosis risk from that pathway, no HERV-K expression, no contribution to certain cancers. This is called individualized virome regulation. The concept, proposed in speculative genomics papers in the early 2020s, envisions a future where each person's endogenous retroviral load is customized, silencing pathogenic elements while preserving those that provide regulatory functions. It sounds elegant. It is terrifying, because we do not know what we do not know. The syncytin proteins required for placenta development, those obviously must be preserved. But what about the HERV sequences flanking the amylase genes, modulating starch digestion? Are they essential or merely vestigial? What about the HERVs near beta globin, fine-tuning hemoglobin production? Could we silence them without causing subtle oxygen transport deficiencies that only manifest decades later? What about the 90% of HERV sequences we consider junk, silent, degraded, apparently functionless? What if some of those sequences are not active now but could be activated under future selective pressures we cannot predict? Evolution does not design for the present. It designs for the range of possible futures. Genetic diversity, including seemingly neutral or deleterious variants, acts as a reservoir of potential adaptations. The sickle cell allele was lethal in non-malarial environments. Then malaria became endemic, and suddenly the deleterious allele was life-saving. The RHD deletion causes hemolytic disease, but under the selective pressures faced by the Basques, it became part of a balanced polymorphism that sustained the population for 35,000 years. If we silence HERVs, if we delete problematic alleles, we are narrowing the range of futures humanity can survive. Should we? Should we edit the RHD gene, restore it universally in Rh-negative individuals, or delete it completely from everyone? Should we silence HERV-W to prevent multiple sclerosis, knowing we may be removing a defense against a pathogen that does not yet exist? This is not a question of capability. We can edit embryos. This is a question of wisdom. Are we intelligent enough to know what we are deleting? Are we humble enough to admit we cannot predict what threats will emerge in 100 years, let alone 10,000? Every decision to improve the genome is simultaneously a decision to narrow the range of futures humanity can survive. Consider the R system in this light. If we could edit the RHD gene, restore it in Rh-negative individuals, or delete it universally, should we? Eliminating renegativity would prevent hemolytic disease entirely. No more stillbirths from maternal-fetal incompatibility. but it would also eliminate the heterozygote advantage. If a novel pathogen emerges, a parasite similar to Toxoplasma or a neurological virus, then DT heterozygotes might be the only genotype with sufficient metabolic resilience to resist it. Delete the D allele, and we lose that option. Now imagine this decision scaled across the entire genome, not just RR, not just HERVs. Every polymorphism, every balance system, every trade-off that evolution has maintained because diversity itself is adaptive. Do we flatten them, optimize for the present, or do we preserve the contradictions, accepting the suffering they cause because we cannot see what they are preparing us for? There is a darker scenario, rarely discussed in public bioethics debates, but whispered in genomic circles. What if different populations make different choices? Some societies may embrace aggressive gene editing, silencing HERVs, eliminating disease-associated alleles, optimizing for intelligence, longevity, physical resilience. Others may reject it on ethical, religious, or philosophical grounds, choosing to remain unidit. Over generations, the genetic divergence between these populations could become substantial, not through natural selection, but through deliberate human choice. And if the edited populations thrive, if they avoid the diseases, live longer, outcompete the unedited, then a new form of speciation could emerge, not by geography or drift, by design. This is not science fiction. This is a plausible trajectory of the next century. And the rice system, the maternal-fetal immune conflict, the balanced polymorphism, the population-level persistence of a deleterious allele serves as a preview. It shows us that evolution tolerates, even requires, genetic incompatibilities. It shows us that what looks like a defect at the individual level may be adaptive at the population level. And it warns us that flattening diversity, even for compassionate reasons, carries risks we cannot fully calculate. Now turn to deep time, not the next 20 years, the next 1,000. If we do nothing, if we allow evolution to continue operating as it has, what happens to the raw polymorphism? The prediction from population genetics models is decline. As sanitation improves, as parasitic infections are eradicated, as nutrition stabilizes, the selective pressures that maintain the heterozygote advantage weaken. Toxoplasma becomes rare in developed nations. Vector-borne diseases are controlled. Zoonotic transmission drops. The fitness benefit of the D genotype diminishes, and the cost, hemolytic disease, even with ROGAM reducing mortality, remains. Over generations, the deletion allele declines in frequency. In 500 years, global Rh negative frequency might drop from 15% to 5%. In 1,000 years, perhaps 1%. In 5,000 years, it might exist only as a rare variant, a genetic curiosity like the CCR5 delta 32 mutation that confers HIV resistance in a tiny fraction of Europeans. The Basques, no longer isolated, intermarrying freely with neighboring populations, will converge toward the European average. The 47.2% frequency will become a historical footnote. This is not tragedy. This is evolution. Alleles that were adaptive under Paleolithic conditions become neutral or deleterious under modern conditions. Selection relaxes. Drift takes over. Diversity erodes. It has happened countless times in human evolutionary history. Lactase persistence arose when pastoralism emerged, allowing adults to digest milk. In populations without dairy traditions, the mutation never spread. Skin pigmentation lightened in northern latitudes where UV radiation was scarce, allowing vitamin D synthesis. At the equator, dark pigmentation was maintained for protection. The Ross system is following the same trajectory. It was adaptive in a specific time, in a specific place, under specific pressures. Those pressures are fading, and with them, the polymorphism will fade, unless the selective pressure returns. or unless we deliberately preserve it. Gene banks. Cryopreserved embryos carrying rare alleles. Genetic diversity archives maintained by international consortia, insurance against futures we cannot predict. This is being discussed seriously by conservationists and futurists. The idea that we might treat human genetic diversity the way we treat endangered species, as a resource to be protected, not because every variant is immediately useful, but because we do not know which will become essential. But there is a philosophical problem here. Preserving diversity in a gene bank is not the same as preserving it in living populations. Evolution requires selection, variation under pressure, adaptation in real time. Frozen genomes do not evolve. If we archive the RHD deletion but allow it to vanish from living humans, we are preserving a museum piece, not a living system. And this returns us finally to the Basque enigma, the question we have circled but never answered. What, specifically, was the selective pressure in the Franco-Cantabrian region that was strong enough to maintain 47% Rh-negative frequency for 35,000 years? We know it was not genetic drift. The frequency is too high, too stable. We know it was not alien intervention. There is no evidence, and the heterozygote advantage model explains the data without invoking the extraordinary. We know it involved balancing selection, where DD heterozygotes had superior fitness under local conditions. But what were those conditions? The answer may be irretrievably lost. Paleolithic pathogens do not fossilize. Environmental toxins do not leave signatures in the archaeological record unless they involve heavy metals or volcanic events. The Basques themselves have no oral tradition explaining their blood. Euskara contains no myths of plague or purification that might hint at the selective force. All we have is the genetic signal, a deletion at 47.2% frequency, a language with no relatives, a population that maintained reproductive isolation in mountain valleys while Europe was reshaped by Neolithic migrations, Bronze Age conquests, Roman occupation, medieval pandemics. Perhaps the selective pressure was altitude, chronic hypoxia in mountain settlements, where optimal oxygen transport, mediated by precisely calibrated Rh protein density, meant the difference between cognitive sharpness and impairment. Perhaps it was dietary. Unique local foods, shellfish, mountain herbs, fermented dairy, that required specific metabolic processing enhanced by HERV-regulated genes linked to the Rh locus. Perhaps it was social. If the Basques practiced endogamy for cultural reasons and the heterozygote advantage was real, then any external population with lower Rh-negative frequency would have been at a competitive disadvantage, reinforcing isolation. Or perhaps, and this is the most haunting possibility, the selective pressure was not one thing, but a constellation of pressures unique to that place and time that can never be fully reconstructed. A specific parasite strain in the local fauna, a specific viral ecology in the caves where they sheltered, a specific ratio of dietary selenium to iodine that affected immune function, a feedback loop between genetic frequency, cultural practices, and environmental conditions that became self-reinforcing. If that is true, then the Basque genome is not a puzzle to be solved. It is a monument to contingency, a reminder that evolution operates in contexts so specific, so local, so entangled with unmeasured variables that reconstructing the full causal chain from genetic data alone may be impossible. We can describe the result. We cannot recreate the experiment. And this is the final lesson for the future scenarios we are constructing. As we design therapies to silence HERVs, as we edit embryos to remove disease alleles, as we debate whether to preserve or eliminate the Rh polymorphism, we are doing so in ignorance. We do not know what selection pressures the next millennium will bring. Climate change, novel pathogens, extraterrestrial colonization, artificial environments where gravity, radiation, and atmospheric composition differ from Earth. The genome we are editing today was shaped for a planet we are rapidly leaving behind. The Basques thrived because they maintained diversity in the face of an unknown threat. Perhaps we should do the same. Not by freezing ourselves in genetic stasis, clinging to Paleolithic configurations, but by ensuring that the diversity we carry, the polymorphisms, the HERVs, the apparently deleterious alleles, is not discarded in haste, because evolution is not a ladder. It is a hedge. A bet on multiple futures simultaneously, knowing that the one that arrives may not be the one we prepared for. In 20 years, temolizumab or drugs like it may become standard therapy for autoimmune diseases linked to HERV activation. In 50 years, individualized virome regulation may be routine, with newborns receiving epigenetic edits to silence pathogenic viral elements. In 100 years, the descendants of edited and unedited populations may look at each other and wonder if they still belong to the same species. And in 1,000 years, someone may sequence a Basque genome from a frozen tissue sample and ask, why did they tolerate so much death to preserve this polymorphism? What did they know that we forgot? The answer is, they knew nothing. They were not wise. They were not prescient. They simply survived. And survival, under conditions we can no longer reconstruct, required carrying contradictions, RO positive and RON negative, predator and prey, viral and human, in the same population, the same family, the same body. We are about to decide whether to preserve those contradictions or resolve them. History will judge whether we chose correctly. But history will not know what future we foreclosed. The future is not written, but the ink, viral, human, hybrid, is ready. The maternal immune system does not malfunction. The ROF deletion is not a defect. The Basque polymorphism is not an accident. These are not failures of biology. They are features sculpted over 100 million years by viruses that integrated into germline cells, by pathogens that selected for diversity, by selection pressures that decided the death of some children was an acceptable cost for the survival of the population. We began with a question that sounded like a violation of natural law. We end with a reframing. The maternal immune response to the R-positive fetus is not a malfunction. It is the inevitable consequence of a genome constructed from negotiation between primate ancestry and viral integration, between genetic uniformity and adaptive diversity, between the preservation of offspring and the preservation of the population's capacity to survive what comes next. The ROS system is a balancing act. DD, optimized for parasites, vulnerable to viruses. D, balanced for both, optimal for neither. D, resistant to viruses, vulnerable to metabolic stress. 3 strategies. 3 futures. 3 bets on what comes next. Evolution does not choose. Evolution maintains all 3. Evolution does not favor one genotype. It favors the coexistence of all 3, because environments are not stable. Threats are not predictable. What protects you against today's parasite may kill you in tomorrow's pandemic. What saves the population in one generation may doom it in the next, unless the population carries enough variation to respond to both. This is why the RHD deletion persists despite hemolytic disease. Not because evolution is cruel, because evolution is strategic. It tolerates individual deaths if they maintain population-level diversity. It sacrifices certainty for adaptability. It writes the genome not as a single perfect text, but as a library of contradictory drafts, each useful under different circumstances. And the library is not purely human. 8% of every page, 203,000 passages scattered across 46 volumes, was written by viruses. Retroviruses that infected our ancestors, integrated into germline cells, and became permanent features of our biology. Some of those passages are blank now, degraded, silent. Others are functional, essential. The syntactin proteins that build the placenta, the regulatory elements that modulate hemoglobin, amylase, immune genes. Without viral domestication, you would not exist. Placental mammals depend on retroviral envelope proteins repurposed for embryonic development. The maternal-fetal interface, the site where ARS incompatibility becomes lethal, is itself a viral invention. We are fighting a blood war across a battlefield built by ancient parasites. This is the synthesis. Humanity is not one species with one genome executing one evolutionary strategy. Humanity is a collection of lineages, each carrying different viral integrations, different deletions, different polymorphisms maintained by different selective pressures. The Basques are not anomalous. They are exemplary. They show what happens when a population experiences intense, localized selection for 35,000 years while remaining reproductively isolated. The deletion reaches 47%. The language diverges into an isolate. The genome preserves a signal from the Paleolithic that exists nowhere else on Earth. We do not know what they were defending against. The pathogen, the toxin, the environmental pressure. It left no fossil, no record. Only the blood remembers. And even the blood does not explain. It only testifies. But we know what they achieved. They survived. They maintained a polymorphism so finely balanced that both homozygotes and heterozygotes persisted, generation after generation, through ice ages and migrations and plagues. Because the diversity itself was the adaptation, now we stand at a threshold. The age of passive inheritance is ending. Gene editing, HERV silencing, individualized virome regulation. We have the capacity to rewrite the viral passages, delete the deleterious alleles, optimize the genome for the present. But optimization is a wager. It assumes we know which future we are optimizing for. The Basques did not optimize. They diversified. They carried incompatibilities, Rh-negative mothers whose bodies attacked Rh-positive children because the cost of those incompatibilities was less than the cost of genetic uniformity. We are about to make the opposite choice, to flatten variation, to silence the viral heritage, to eliminate the polymorphisms that cause suffering. Perhaps we are right. Perhaps modern medicine has rendered ancient adaptations obsolete. Perhaps we can afford to narrow the range of human genotypes because we have technology to compensate for whatever threats emerge. Or perhaps we are wrong. Perhaps in 1,000 years when a novel pathogen emerges, or when humans colonize Mars and face radiation pressures Earth never imposed, the descendants of the unedited populations, the ones who preserve their viral load, their polymorphisms, their contradictions, will be the ones who survive, and they will look back at us and ask, "Why did you delete the insurance?" We cannot answer because we do not know. The future is opaque. All we have is the past, written in blood and viral code, testifying that diversity itself, costly, contradictory, lethal to individuals, has been the strategy that carried us from primate ancestor to global species. The Ross story is not about blood types. It is about what it costs to remain adaptable in a universe that does not announce its threats in advance. It is about the trade-offs embedded in every cell, every pregnancy, every generation. It is about the fact that the human genome is not a blueprint. The human genome is not a blueprint. It is a treaty, signed in viral ink, written over primate parchment, negotiated between what killed us and what we refused to let die. And the raw polymorphism is one clause in that treaty, paid for in children's lives, renewed every generation for 35,000 years, maintained because the alternative was not individual suffering but population extinction. It is a negotiation between virus and host, between mutation and selection, between the individual who suffers and the population that endures. The maternal immune system that attacks the R-positive fetus is not broken. It is executing an instruction written 35,000 years ago, in response to a pressure we can no longer name, for a purpose we can only infer. It is doing what evolution trained it to do: recognize difference, defend boundaries, maintain the tension between self and other that allows populations to carry multiple solutions to problems not yet encountered. Some children die. This is the cost. The population persists in its contradictions. This is the strategy. And we, standing on the far side of medical progress, able to prevent those deaths, must decide whether to pay the cost or abandon the strategy. There is no neutral choice. Every decision rewrites the future. The synthesis is this: we are not invaded by our past, we are composed of it. Viral and primate, compatible and incompatible, homozygous and heterozygous. And the maternal-fetal blood war is not a medical error to be corrected. It is evolution reminding us that survival is not about perfection. It is about maintaining the capacity to become something else when the world demands it. Return to the beginning, to the moment, quiet, invisible, occurring in the darkness of the womb, when a mother's body makes a decision. We understand it now differently than we did 2 hours ago. The antibodies crossing the placental barrier are not malfunctioning. They are reading the genetic signature, the D antigen, present on fetal cells and responding exactly as millions of years of evolution calibrated them to respond. Recognize difference. Assess compatibility. Defend. Place your hand on your chest again. 70 beats per minute. Blood pushed through arteries. Delivered to tissue. Returned to the heart. But now you know it is not just your blood. It is primate blood. Viral blood. Basque blood. Balanced blood. Written on every red blood cell is the memory of negotiations you did not make, defenses you did not choose, contradictions you inherited because your ancestors could not afford genetic certainty. The tragedy is that the fetus is not an invader. The tragedy is that the genome itself is divided, RRRA positive and RA2 negative lineages coexisting in the same species, the same valley, the same family. Because division was adaptive, because maintaining both variants, despite the blood incompatibility, provided a survival advantage that no single genotype could match. The mother's immune system is not attacking her child. It is defending a boundary that evolution drew 35,000 years ago in response to a threat we can no longer see. A boundary that cost children then, a boundary that costs children now, but a boundary that, for reasons encoded in Basque blood and viral ancestry, has never been allowed to collapse. We are all boundary creatures, viral and primate, self and other, compatible and incompatible. The human genome does not belong to a single species executing a single strategy. It is a palimpsest, a library, a negotiation that has been ongoing for 100 million years, since the first retrovirus integrated into a mammalian germ cell and became hereditary. 8% of you is viral code. Some of it builds the placenta that allowed you to develop inside your mother's body. Some of it regulates the hemoglobin that carries oxygen through your blood. Some of it lies dormant, waiting for a trigger. Inflammation, infection, stress, to reactivate and potentially cause disease. You are not invaded by viruses. You are composed of them. And the raw polymorphism, the deletion that defines RA negative blood, the heterozygote advantage that maintains it, the maternal-fetal conflict that punishes it, is one thread in a larger tapestry. A tapestry woven from contradictions maintained because contradictions are how evolution hedges its bets. The Basques stand as witnesses, not to alien intervention, not to lost civilizations, but to the power of isolation selection, and time to sculpt a genome into a configuration so extreme that nearly half the population carries a deletion that should, by every conventional model, be extinct. They carry it still. Not because they are wiser, because they survived. And survival, in evolutionary terms, is the only argument that matters. Not the argument of comfort, not the argument of fairness, the argument of what persists. We do not know what selective pressure created the Basque ratio. We may never know. The pathogen left no skeleton. The environment left no monument. Only the blood remains, testifying in a language we can read but not fully translate. But we know this. Whatever the pressure was, it was strong enough that evolution decided the death of some children was an acceptable cost for maintaining the diversity that protected the population. We know that 35,000 years later, the polymorphism still stands. We know that Euskara, the Basque language, still has no relatives, as if the people themselves are linguistic and genetic relics of a trajectory that diverged before recorded history. And we know that in the valleys of the Franco-Cantabrian region, in the caves where Paleolithic artists painted aurochs and left handprints on stone, there is a silence. The silence of questions that have outlived their answers. Tonight, somewhere, a woman is giving birth to her second child. She is RE negative. The child is RE positive. Her immune system, primed by her first pregnancy, is deploying antibodies. Modern medicine will save that child. ROA. Exchange transfusions. Neonatal intensive care. We have rendered the ancient conflict survivable, but we have not erased it. The antibodies still form. The incompatibility still exists. We have overridden evolution, not rewritten it. And someday, perhaps in 50 years, perhaps in 500, we may choose to rewrite it, to edit the genome, to delete the polymorphism, to resolve the contradiction. Or we may choose to preserve it, recognizing that we do not know what futures we are optimizing for. And diversity itself may be the most valuable inheritance we can pass to the generations not yet born. There is no conclusion to this story because the story is still being written. In clinical trials, in gene editing labs, in Basque villages where children are born carrying blood that remembers pressures no living human has faced. The maternal kill switch is not an error. It is a reminder that the human body is not a machine. It is an archive of negotiations between host and parasite, self and other, individual and population. A reminder that what looks like violence may be defense, that what looks like defect may be adaptation, that what looks like suffering may be the cost of remaining adaptable in a universe that does not announce its threats. We are not one species. We are a collection of lineages, viral and primate, compatible and incompatible, held together by selection pressures we can measure but not always name. The null lineage is not missing. It was never null. It is present in every drop of Rh-negative blood, every heterozygote balancing contradictions, every polymorphism that refuses to resolve. And in the mountains between France and Spain, where a language unrelated to any other is still spoken, where the deletion reaches frequencies seen nowhere else on Earth, the Basques endure, not as aliens, not as relics, but as humans who carried a contradiction long enough for it to become identity. The genome is a palimpsest, viral ink over primate parchment, deletions inscribed over duplications, conflicts maintained over resolutions, and every child born is a new page, carrying forward the contradictions their ancestors could not afford to eliminate. In the beginning, we asked, why does the human immune system possess a specific, encoded instruction to attack its own progeny? The answer is, because evolution does not design for peace, it designs for survival. And survival requires carrying the tools for war, even when the war is between mother and child. Even when the enemy is written into the blood itself, we are not invaded by our past, we are composed of it. And the signal in the blood, Rh negative, heterozygous, incompatible, is not noise, it is memory. Genetic memory of pressures we no longer face, adaptations we no longer need, conflicts we can now prevent but not erase. The maternal immune system will continue attacking Rs-positive fetuses for as long as the deletion exists. Evolution wrote that instruction 35,000 years ago in response to a threat we can no longer name. We have learned to interrupt it with medicine. We have not learned whether interrupting it was wise. The future will answer. But the answer will not come from laboratories or gene editing consortiums. It will come from the blood itself, from which lineages persist when the next pathogen emerges, which deletions survive when the environment shifts, which contradictions prove adaptive when the threats we cannot predict arrive. In the darkness of the womb, a decision is being made, not by conscious minds, by molecules, by antibodies, by genetic instructions written before we had language to name ourselves. The decision is the same now as it was 35,000 years ago. Recognize, assess, defend, and in that decision, violent, ancient, inevitable, we see what we are, not masters of our biology, inheritors of it, carrying contradictions forward because contradictions are how life survives what it cannot predict. The blood remembers, even when we forget.